Hello everyone, my name is Rahul. In the previous video, we had discussed about polymerase chain reaction. In this video, we are going to discuss about insertion of recombinant DNA into the vector. So, the ligation of DNA fragment into a vector an enzyme, requires an enzyme known as ligase. Requires an enzyme known as ligase. So, the transfer of recombinant DNA into the vector, it requires that the complementary sticky ends of passenger and vehicle DNA are joined together with ligase enzymes. So what we do is, first we cut the required DNA fragment, our gene of interest, with a polymerase, with a endonuclease, and the vector DNA will also be cut using the same endonuclease. So required gene of interest and the vector DNA are cut using same restriction enzyme. So what this does is, it produces same same type of sticky ends in both required DNA and vector. So for example, if we are cutting both the required DNA or our gene of interest and the vector with eco R1, so it will produce the same type of sticky ends, and when we will put these two DNA fragments into a medium, and in the presence of enzyme ligase, the sticky ends, because they are complementary, will ultimately form the hydrogen bondings. The sticky ends will attach and form the recombinant DNA. So by cutting both the gene of interest and the vector DNA by a same restriction enzyme, we are essentially producing sticky ends, because these sticky ends are being formed by the same restriction enzyme. They will be complementary to each other, and they will stick to each other and attach, and the recombinant DNA will be formed. Now this recombinant DNA, it has to be transferred into the host. This is known as transformation. The details of transformation we have already discussed in the previous videos. So to summarize, gene transfer is achieved by two kinds of transfer methods. First is indirect methods, and the other is direct methods. In the direct methods, we have electroporation, by using chemicals, by treatment with divalent cations such as calcium, microinjections, gene gun, or biolistic methods. So these are the methods, or liposome mediated transfer. So these are the methods which are known as the direct methods of gene transfer. In indirect methods, we use of vector. A vector may be a bacteriophage, it may be a plasmid, it may be a cosmid, it may be a adenovirus, it may be a retrovirus. So the details of all methods of transfer we have already discussed in the previous video. Now after the process of transformation, to check whether the cell have taken up the gene of interest, we perform selection of transformants, which is done by two techniques. First is selection using antibiotic resistance genes, and the other is by using lac Z gene and one antibiotic resistance gene. The details we have already discussed. So to summarize, what we do is we ligate the foreign DNA at the antibiotic resistance site. For example, we can ligate the foreign DNA at the BAM-H1 site of tetracycline resistance gene in the vector PBR 322 that we have already discussed. So when foreign DNA inserted into BAM H1 site of PBR 322, there will be insertional inactivation of this tetracycline resistance gene. So what we will do is we will grow these cells on a medium containing tetracycline. Transformants will not be able to grow on medium containing tetracycline. But to be sure, to confirm this, what we do is first we transfer the cells on a medium containing ampicillin. Then we transfer the cells on the medium containing tetracycline. So the transformants, because we have not done anything to the ampicillin resistance gene, the transformants and the non-transformants, they will both grow on the medium containing ampicillin. But as soon as we will transfer these cells to the medium containing tetracycline, the transformants will not be able to grow and the non-transformants will keep on growing. So we will select the transformants and we will use these cells to further multiply and get our desired product, which we will discuss later. Now selection using the LAC-Z gene. LAC-Z gene is responsible for the production of an enzyme known as beta-galactosidase. So this LAC-Z gene is responsible for production of an enzyme known as beta-galactosidase. Beta-galactosidase, what it does is, in the presence of chromogenic substrate, when we add chromogenic substrate to the cell, this beta-galactosidase leads to formation of a colored product. So when we will attach our gene of interest onto the LAC-Z gene, what it will do is insertional inactivation of LAC-Z gene. This beta-galactosidase will not be produced and this colored product will not be formed. And this is how we will identify our non-transformants and the transformants. The transformants, this colored product will not be formed, not produced in transformants. So, and to confirm, we can also use the antibiotic resistance simultaneously. We will transfer these onto the medium containing at least two antibiotics such as both ampicillin and tetracycline. Our transformant should be resistant to both the tetracycline and ampicillin because we have not done anything to the genes containing ampicillin and tetracycline. So this is how selection is done for transformants and the non-transformants. Now to summarize the steps of recombinant DNA technology, what we do is we take a foreign DNA which has the gene of interest, we take a vector DNA which may be a plasmid, a cosmid or any other such vector. We will cut both the vector DNA and the foreign DNA using the same restriction enzyme producing the sticky ends. Then we will use the enzyme ligase. This ligase will attach our foreign DNA into the vector. This ligase will attach our foreign DNA into the vector. So now we have the recombinant DNA molecule. This recombinant DNA molecule is inserted by a process known as transformation, which can be indirect or direct. Once this is inserted into the host cell, these cells will divide and our DNA is keep on increasing. So from 1 we will have 2 cells, from 2 cells we will have 4 cells, and our required gene, it is present in all these cells. So from 1 gene we have now 4 genes. Similar is the case in PCR. So instead of doing all this, we can simply perform the PCR to get more number of genes to amplify the genes. But in case of cells, this is how gene amplification is performed. With this video, we finish off with our discussion on recombinant DNA technology. In the next video, we will discuss about obtaining the foreign gene product, which is our product of interest. See you in next video.